Hello everybody, hope you are all okay. So today I will teach you the last chapter of this course, the DNA, about the DNA structure and its function and RNA. DNA and RNA play an important role in the cell, in the living organisms. So DNA and RNA structure and function. Okay, this chapter DNA, chapter 11 is a big chapter which needs about 2-day class. So I started recording. Today I will record the first part of the structure and function of DNA, this DNA chapter 11, and if I can finish the whole class, it's fine. Otherwise, I will make another session and recording the part 2. So this is the first part. So DNA is the genetic material. That means DNA has the gene, and this gene carry all the genetic material from generation to generation, like from our parents to us, like my color, my skin color, my eye color, my behavior, my characteristics. So all of these things we usually get from our parents. How we get it? We got it through this DNA, and the gene carry this DNA. So it carry all of the information of the characteristics of living beings. Now, once the scientists know that yes, DNA is play an important role in the living organisms, then they want to know what is the structure of DNA. Because if the scientist can discover the structure of DNA, then they can play with it and they can do so many things with this DNA structure. So in the world, in different places, in different labs, in different countries, the scientists started like race to determine the structure of DNA. But here we are discussing some of the important discovery, partially who discovered partially the structure of DNA. First one called Chargaff's rule. Erwin Chargaff was a scientist who was also tried many years before to determine the structure of DNA. So what was his discovery? He already knew that DNA has 4 types of nucleotide. What are these 4 types of nucleotide? These 4 types of nucleotides, I think everybody should know that. One is adenine, then guanine, cytosine, and the last one is thymine. Adenine, thymine, guanine, and cytosine. So Chargaff knew that DNA contained these 4 types of nucleotide. So adenine we called A, adenine is A, then guanine is in other places I will write. Guanine is G, cytosine is C, and thymine is T. A, T, G, and C. So he found these 4 nucleotides and then he examined many DNA from many species. And what is other information he got? The first information, important one, is the amount of A, T, G, and C. Amount of A, T, G, and C in DNA vary from species to species. Like in a human being, we have huge number of species present, million trillions of species are present, and like in this classroom, we have suppose 30 students. Each of us are different from each other. Maybe you are different from me, I am different from the other students, she is different from the other students. So which point we are different? Because all of we are human beings. We should be, everything should be same in our body, but not. We are different in different skin color or eye color or hair color or in behavior. So in many cases we are different from each other. So which point we are different? We are different in these 4 points, 4 nucleotide. So the amount of A, T, G, and C in the DNA vary from species to species. What it means? It means that if I have this adenine, A adenine is 25%, suppose, it does not mean that all of you, all of the other students in my class, 30 students, all of them have the same 30%. No, it is different from each other. Like maybe you have 30%, the second student has 28%, the third student has 35%, the fourth student have 40%. This amount is variable, vary from species to species. That makes each of us different from each other. Okay, so the amount, it is very important to understand that amount of A, T, G, and C vary from species to species. The second point, in each species, like I am one of the species, one student is another species, third student is another species, fourth student is another species. So every species is different, although we are all human beings. So in each species, the amount of A equal to T. What it means? It means that if I have A is suppose 28%, that means I have the T is also the same 28%. If A is 28%, I have T is also 28%. So this is the meaning. If suppose if I have G is 25%, amount of G is always equal to C. That means C is also 25%. Now I am writing down in another let me go to the whiteboard. Okay, suppose if I have A is 25%, then what it means, I have G also, not G, if I have A is 25%, A is always equal to T. So that means T is also 25%. Okay, now what will be the amount of G and C? Can you find out? Because the amount of A, T, G and C, all of them together is 100%. A plus T plus G plus C is equal to 100, is a percentage. So in this percentage, if A is 25% and T is 25%, that means 50% is gone. So the rest 50% is equally divided between G and C. So how much the G get? The G get 25 and C also get this equal amount 25. So it means if I know the amount of A is, I can easily find out the other three nucleotide percentage. This type of question also can come in the final exam. Suppose the question can come in a nucleotide if the amount of A is 35%. Okay, 35%. Then what is the amount of C will be? C will be. What is the amount of C will be? Can you find out? Okay, let me explain. If A is 35, that means G is also equally 35. So A plus G, A equal, not G, I am sorry, I made the mistake, T. A is always equivalent to T, so T is 35%. So A is 35%, T is 35%. A is 35% and T is 35%. That means 35 plus 35, 70% gone. Then how much left? Only 30%. That 30% is equally divided between G and C. So 30 is divided by 2 equally. So equally means G will get then 15% and C will get 15%. So that means if I know that if one nucleotide A is 35%, can I know what is the other three nucleotide will be? Yes, I can know because A is always equal to T. So T is 35% and the rest 30% divided equally between G and C, which is 15 and 15. So this is the second important information. So what is Chargaff's rule? He knew that the amount of A, T, G and C variable vary from species to species, and second is the amount of A is equal to T and G is always equal to C. You also, another information he told that all the nucleotides contain three things. One is a phosphate group, a 5-carbon sugar and a nitrogen containing base. So these already you know what are the three components of a nucleotide. So this is the each nucleotide, one nucleotide, a phosphate group, a sugar, 5-carbon sugar and any one nucleotide. Then another scientist, Rosalind Franklin. She was a scientist who was working in London, King's College of London, and she also studied the structure of DNA. Now, what was her discovery? Her discovery showed that DNA to be a helix. That means DNA is a helical structure. DNA is a helical structure, like this is one DNA is double stranded, this is one strand and this is another strand, make a helix. So DNA to be a helix and some portion repeating over and over. So that means this portion may be repeating in here, this portion may be repeating in here, over and over. And how she found this result? She followed a method, she did a method which is called X-ray diffraction data or diffraction pattern. So maybe the question can come in the test that what is Rosalind Franklin discovery? Her discovery was, the answer is, her discovery showed that DNA is a helical structure with the some portion repeating over and over. Another question can come, how she find out this discovery? She find out this discovery by follow a method called X-ray diffraction pattern. Okay, then finally two scientists, James Watson and Francis Crick, they gather all these previous information and finally made the model of DNA. And because of this discovery, they won Nobel Prize in 1951. Okay, so these two scientists finally discovered the model of DNA. This model also explains how the replication process works. That means how the DNA become duplicated, become double from one DNA to two DNA, from two DNA to 4 DNA. And replication process are used in the cellular reproduction. Remember, try to read the chapter 8 again, that before the cell division starts mitosis, the DNA or gene the chromatid become duplicated, but they are joined in a place called centromere. So this duplication process done by replication. So this model also explain how the replication process works. And this model still used today with very few small changes. Okay. Then how they made or build up this model, the DNA model? They made this model by wire and pin. So they use wire and tin to build up this model. So that's why their model also called wire and tin model. Their model also called wire and tin model. Then how they explain the DNA structure? They explain the DNA structure that it is a double helix. That means DNA is a strand, has two strands. DNA has two strands. These are the two strands of DNA, and like a twisted ladder. So if a ladder has two sides, but this one is become twisted. Like if I draw like this way, then one point they become twisted. It is like a ladder, but they become twisted. Okay, now the sugar, which is a 5-carbon sugar. For DNA, this sugar called deoxyribose sugar. It is also important question can come. What is the name of the sugar for DNA? So the name is deoxyribose sugar. So this sugar and phosphate, they are become bonded, and where they are present, they forming the side. Side means in this part. They forming the side, that means in this place. And with the bases, base is 4 nucleotide, adenine, guanine, cytosine and thymine. They making up the rungs of the ladder. You can see in the next picture in here. This sugar, 5-carbon sugar and phosphate are present in the side of the DNA, and the bases adenine, guanine, cytosine and thymine, they are present, make the rungs of this ladder. Also, they follow the previous scientist information, Erwin Chargaff's rule, that A is always paired complementary with T and G is always complementary with C. It is also important. A amount of A is equal to T, G is equal to C. Okay. Then come this base pairing called complementary base pairing of A and T, G and C. Then how these A and T, G and C, they are joined or bonded. They are joined by hydrogen bond. You can see in here the hydrogen bonding between the bases hold the half of the helix together. So they are joined by hydrogen bond. So, and another information is that A is always paired or make bonding with T by 2 hydrogen bond, and G is bonded with C by 3 hydrogen bond. So A made bonded with T by 2 hydrogen bond, and G always bonded with C by 3 hydrogen bond. It is important to remember, question can come. Okay, so when they know the reapplication of DNA, then what they do, they start to know how this DNA become double or duplicated. So replication of DNA. What is the definition of the replication of DNA? It is the process of copying of DNA before the cell division. So before the cell division, when the DNA become double from one chromatid to two chromatid, but they are joined in a place called centromere. So this process called replication, replica of DNA, exact copying of the original DNA. Okay, how they do this? things, replication process occurs. This process, the replication process, called semi-conservative. Semi means partial and conservative means conserve. So, what is the meaning? Each new DNA molecule is made from one parent strand and one new strand. I will show you a picture. You can see the picture here. This is the original two strand of DNA. The left one is one strand, other one other strand. Then these two strands become separated. One is going in this left side, the other strand going in the other side, right side. When they start to become separated, then these base A, T, G and C, they also become separated. Like you can see in the picture, one nucleotide going in with one strand and this nucleotide going in the other strand. So this is the one separation. So now in here, each new DNA molecule is made up of one parent strand and one new strand. I will explain it in the picture later on. So how did replication process occurs? It requires three steps. First step called unwinding. Okay, unwinding means unzipped. Like in when we are wearing a jacket and zipped up, then zipped up means both side, the right side and the left side of the jacket, they become bonded, joined together. And then when we unzipped them, that means opened up them, we separate these two, the zipper, right side and left side. In here also the two strands first unwinded or unzipped, that means separated. So this separation is not done instantly. This separation is done by an enzyme. So you should know the name of the enzyme. So this unwinding process is done by helicase enzyme. You have to remember the name of the enzyme because question can come that what is the name of the enzyme for unwinding. Okay, so that means they become separate now, the two strands. When these two strands become separate, then what happened? Like from the beginning, the two strands are in here. One is going in this side and the other one is going in this side. They become separate. Okay, then when they become separate, the A, T, which are joined in the middle, they also become separate. So maybe A is going this side, T is going in the other side. So, but it is important that according to Chargaff's rule, these nucleotide should be complementary to each other, should be stay together. So A cannot stay without T and G cannot stay without C. So, but in here when they become separate, they become alone. So, they cannot do any work. They need their complementary bases. So, then the second thing happened called complementary base pairing. So, what happened in here? So, in this point, when they become separate, from here the new strands start to produce. Like you can see in another color, like if there is A here, then T will be in here. The same way in here also, the new strands start to produce in here with the complementary pairing. So this is called complementary base pairing. But here is a mistake, mistake printing. Complementary base pairing is done by DNA ligase. I am writing down in here. Complementary base pairing is done by DNA ligase. Okay. Then the third one. Now third one called joining. The joining means now this strand, this black one, this strand joined with the blue strands, the original one, and make a new DNA. This is one daughter DNA and this is another daughter DNA. So this joining is done by, oh, it's not, I made the mistake here. So okay, this complementary base pairing is done by DNA polymerase. Okay. Now the joining one. So this is not the correct one. The complementary base pairing is done by DNA polymerase, and then when they become joined, this is joining is done by DNA ligase. So you should remember the third orderly, the three steps of the DNA replication. First one is unwinding, next one complementary base pairing, and third one is joining. Also remember the name of the enzyme. Unwinding process is done by helicase enzyme, complementary base pairing is done by DNA polymerase enzyme, and joining is done by DNA ligase enzyme. Now, when these two DNA, daughter DNA are produced, they call daughter and the original one called parent. Like in our family, our parent before is the first generation parent, and from there the children comes, the children's called daughter and son. So this in here scientists they use the word daughter DNA. So the new DNA molecule is exactly identical to the original parent DNA. So you can see here this is daughter DNA, this is another daughter DNA, and these two daughter DNA are exactly identical to their parent DNA. Now the whole process you can understand. The semi-conservative replication. The parent strands unwind or unzipped first as separated by the action of helicase enzyme. Then the new strand form through the complementary base pairing. How? By the action of which enzyme? DNA polymerase. Then the third steps, DNA ligase seal any break in the sugar phosphate backbone, and they joined the sealing, the two strands. Then we get a new daughter, new daughter DNA. So this DNA molecule, the new one, will be half old. Old means half comes from the parent and half is new. That's why it is called semi-conservative. That means one of the daughter parent strand become conserved in the new daughter DNA. Okay, so and the new daughter DNA molecule will be exactly identical to the original parent DNA molecule. So this new daughter DNA exactly identical to the original parent DNA. That's why this process called semi-conservative replication. Then once the scientists know the structure of DNA and the function of DNA, now they want to know another things, that is RNA. RNA, another nucleotide called RNA, because RNA is another important nucleotide which are used to make a protein, and through this protein all of the genetic information is expressed. So RNA structure. What is the RNA structure? RNA is another type of ribonucleic acid, and you also have some idea about the RNA structure. It contains the sugar, 5-carbon sugar, but the name is ribose sugar. You have to know these things because question can come. What is the name of the sugar for RNA? Ribose sugar. RNA also has 4 bases, but there is one difference. RNA has 4 bases, but it has adenine, cytosine, guanine, and one extra. Instead of T, it has, instead of T, RNA has U or uracil, another bases. And in here, adenine A is equal to U and G is equal to cytosine C. So this is the difference. Instead of T in RNA, adenine is equivalent to uracil, U. RNA is single-stranded. It has only one strand, and RNA are mainly 3 types. We should know them. First one called messenger RNA. But what is the function of RNA? RNA are used for protein synthesis. So try to remember what we learned about the protein. In chapter 3, you also answered the quiz, what is the primary structure of protein? How a protein is made up of? The protein is made up of many amino acids joined together by dehydration reaction and by a peptide bond. That's why when many amino acids joined together, they make a polypeptide. Poly means many. So polypeptide, which is a protein. So these 3 types of RNA are used for this protein synthesis. The first RNA called messenger RNA. What is the function of messenger RNA or mRNA? Then we will know what is the other type of RNA, second one called transfer RNA or tRNA. The third one called ribosomal RNA or rRNA. It is important that all these 3 types of RNA are produced from DNA. That's why the DNA, when the scientists know the DNA structure, when the scientists know this DNA structure, let me write it down. When the scientists know the structure of DNA, then from there they start to produce these 3 types of RNA. The messenger RNA, tRNA, messenger RNA, tRNA and rRNA. Why? Because these three types of RNA are help to make a protein. Okay, now what is the function of each of these RNA? So this is the structure of RNA. You can see RNA is single stranded, and like the DNA, the phosphate and sugar is present in the side and the bases are present in here, and instead of T, RNA has U. So A, U, G and C, these four bases. So you can get the differences between DNA and RNA in here. Now these three types of RNA. All these three types of RNA are produced from DNA. Okay, it is very important. So you can see here now messenger RNA or mRNA. It is produced in the nucleus from the DNA template. Okay, and what is the function? Function is it carry the genetic message to the ribosome. Now, ribosome is an organelle of the cell. Everybody try to remember what is the function of ribosome. The function of ribosome is protein synthesis. That means ribosome is a place, is a heart-shaped structure. You can see ribosome is a heart-shaped structure in the cell, which is a red color in the dotted part in the cell. This is the ribosome where the protein synthesis occurs. That's why the question also came, what is the function of ribosome? The function of ribosome is protein synthesis. So this is the place where the protein synthesis done, and this mRNA, messenger RNA, carry the genetic message to this ribosome. Okay, next is second one, tRNA or transfer RNA. This tRNA also produced from the DNA. And what is the function? It transfer the amino acid to this ribosome. So it transfer the amino acid to the ribosome. Now you have to remember, I think the question also you answered, how many amino acids are used for protein synthesis? I think everybody should know how many amino acids are used for protein synthesis. Can you remember how many? 20. 20 amino acids are used for protein synthesis. So this tRNA, each tRNA carry one amino acid, okay, to the ribosome. That means how many tRNA we needed? We needed 20 different tRNA to transfer these 20 amino acid to the ribosome. So each type carry only one type of amino acid. That's why 20 different tRNA carry 20 different type of amino acid for protein synthesis. It is important. Third one is called rRNA or ribosomal RNA. This RNA also produced from the DNA template, and this RNA basically help to join to produce the ribosome structure. So these are the three types of RNA we should know now. You can see the structure. This is the picture of the ribosome in the cell, the red spot, and this red dotted spot is this ribosome. Once we see them under the microscope, it is this bigger shape. You have to remember the structure of the ribosome. This is the heart shape of ribosome structure, and this ribosome has two unit. Okay, the large part called large subunit, and the lower bottom part is called small subunit. So these are the two units. and the messenger RNA who carry the message, they usually go in between the large and small subunit. Okay, now this large subunit has also two sides. Side means two part, like a two door. If it is, we are thinking as a room, so it has two door. One is this door, another one this door. So this is in here called side. So this is the A side, or we can say enter door. That means through this A side, the tRNA, this is the tRNA, entered into the ribosome. And when this tRNA enter into this ribosome, this tRNA carry, each tRNA carry one amino acid on its head. You can see in the picture, this picture, one tRNA enter and it bring one amino acid on its head. So in which site they enter? This tRNA enter through A site. So through this A site, and then they move forward from, so they enter in this site, then they enter through this site, then they move forward, and then they exit, go out. So this go out exit site is called P site. This exit site is called P site. So this tRNA enter, then move forward, and then they exit. Exit through P site. And so this is the way the tRNA enter, they move forward in the large subunit of ribosomal RNA, and then they enter. Now tRNA, how it looks like. The tRNA is a clover shaped structure. So this is the structure of each tRNA, and I already told you that how many tRNA are used for protein synthesis. 20 tRNA. Why 20 tRNA? Because each tRNA, one tRNA carry one amino acid, and this one amino acid joined with another amino acid, joined with another one, joined with another one, and then this way they make a protein, polypeptide. So you can see in this picture peptide or a protein. Okay, so each tRNA carry one amino acid on its head in here. But what is in the other side, in the bottom side? In the bottom side, they carry anticodon. Okay, now comes what is this anticodon is. Now I am coming the another part called mRNA or messenger RNA. I told you what is the function of the messenger RNA. It carry the message of DNA. Message means how they carry the message. They carry the message as a code word. Like once we write a letter, we got the message through the letter, different type of message, right, in our life. Now, who send us or who carry this message to us? The mailman, right? The mailman carry the message. They left the different letter, different messages in our mailbox. And then once we open this letter, we get different message, right? Now, what is written on different letter? How they write down this letter? They use different word. First they write down different sentences, and how each sentence is made up of. Each sentence is made up of what letter. It can be 2 letter, it can be 3 letter, like how are you is 3 different word, 3 different word. So how are you, it makes a message that how are you doing, right? So same way in here, the messenger RNA carry the message as a code word. Code like HOW, how is a word. The same way messenger RNA also carry the message, that message called codon. Codon or genetic code. Each code carry one message. So now comes about the messenger RNA. Okay, let me explain you the messenger RNA first. Okay, the messenger RNA information in here. The code. The messenger RNA carry the message as a code, and how this code is made up of. Like if the messenger RNA carry the message, like make a sentence, and each sentence is made up like how are you, how is one word, and how it is spelling HOW. So that means 3 letter is used in here. So same is in here. They made a code in here, and each code word is made up by 3 nucleotide, any 3 nucleotides. So this is one code, then this is another code, this is another code, then another 3 nucleotide make one code. Now what is this 3 nucleotide? Let me, like it can be A, then G, then C. The next one is maybe U, G, G. So this way, so like A, G, C is one code, then GCC another code, AAA another code, UGC another code. So this way they make different code. And where are these code is present? They are present in messenger RNA. These code carry the genetic message. Each code called genetic code or codon. It is also called codon, genetic code or codon. Okay, so the each code or codon is made up of 3 bases, any 3. It can be A, G, C, GCC, AAA, UGC, like this way. So these codon are present in the messenger RNA, and because each code is made up of 3 nucleotide, that's why they also called triplet code. Triplet means 3, made up of 3 nucleotide, and each code are used for one amino acid. So this code carry the message of one amino acid, this one carry the message of another amino acid, this one carry and the message of another amino acid. So now when they start to carry the message, the mRNA start to carry the message from here to here, from this side to this side, they start to carry the message. So how they start to carry the message? Now I told you who gives us the letter, the mailman, who bring the different type of message by letter, right? So when the messenger and the mailman start to deliver the mail or the message to us, how the mailman know when his duty will start to deliver the message? Of course, that mailman know from his office that yes, from the office he got an instruction that maybe 10 o'clock you will start your job. What is your job? To deliver the mail in different house. So then at 10 o'clock he start to do his job to deliver different mails in different houses. Same in here. The messenger RNA, when they start to carry this message from this side to other side, they should get an instruction. How the messenger RNA get this instruction? Like the mailman got the instruction from his office. So in where is the office of this messenger RNA? The messenger RNA's office is, there is a start codon, a specific codon, okay, for messenger RNA. What is this codon? This codon called AUG. So when the messenger RNA get this A, U and G, then the messenger RNA understand, yes, now I have to move forward. Now I have to move forward to start to carry the message. Okay, so this is called start codon, A, U and G. So when the messenger RNA gets this AUG, the start codon, the messenger RNA start to carry the message. The same way, when the messenger RNA stop, like when the mailman finished his job, the same way he got the instruction from his office that maybe at 3 o'clock you have to stop to deliver the mail. Same in here also. When the messenger RNA get the instruction that he has to stop it. So there are, the same way, there are 3 stop codon. 3. It is important. 3 stop codon present: UAA, UAG and UGA. When the messenger RNA get any one, either this one or this one or this one, any one of the stop codon, then messenger RNA stop there and tell that yes, my job is done. I cannot deliver more message today. I am stop here. So that's why this codon called stop codon. So remember, the start codon is only one codon, but there are 3 stop codon. When the messenger RNA get any one of these 3 codon, messenger RNA stop to move forward. So it is very important to understand how the messenger RNA start to carry the message by start codon, and how the messenger RNA stop when the messenger RNA get any one of the stop codon. Okay, so this is one important information. Now, it is also important that when the messenger RNA carry the message, different message, AGC, GCC, this message, these are the nucleotide and they cannot move alone. They cannot stay alone. They need their complementary one, another one. So who is the complementary of this A is? Who is the complementary of this A? The complementary of A is U. So the complementary of A is U, then complementary of G is C, then complementary of C is G. So this A complementary with U, G is need their complementary C and C needs the complementary G. Now who brings these complementary codon? This codon called anticodon. Anti means opposite of the codon. Now, who brings this anticodon? So now we are going back and I am showing you who brings this anticodon. The tRNA, remember I will show you again that the tRNA brings this anticodon. This is the tRNA and you can see the bottom there are 3 codon. These 3 codon called anticodon is this one. So each tRNA brings this anticodon is U, C and G. Okay. Now this codon, from these 4 nucleotide we can make million number of different codon. It does not mean necessary that it should be always GCC. It can be any three nucleotide make one codon. So we can get millions of codon from these 3-4 nucleotide A, U, G and C. Okay. So this genetic code is universal. It is another important information. That means genetic code for animal, for the plant, for the bacteria and virus are same. So this is the function of messenger RNA. Now we are going back to this picture. So you can see the tRNA in one side it brings the amino acid on its head and the other side it brings the anticodon. And who brings the codon? This is the one, the messenger RNA carry the codon. Like you can see here, 3 nucleotide make one codon. ACC, AAA. These are different codon or code word. So we are done for this part 1, and next part I will show you how these 3 types of RNA, the messenger RNA, tRNA, rRNA, they are all used to express the protein, to make a protein or helped protein synthesis in the other part called part 2. So we are finishing here for today. So in next again I will in part 2 I will explain how we get these 3 types of RNA are used for protein synthesis.